Okay, welcome to our webinar today, everyone. My name is Ron Myers. I'm the Director of Editorial here at Jove. So for those of you not familiar with Jove, we're a PubMed Index video methods journal. So our host today, Eileen McClellan, she published with us previously on a judgment bias protocol in mice. So today she's going to be updating us on the protocol, telling us if there's been any recent updates, kind of comparing and contrasting with related methods. I will say that we're recording the webinar, so we'll send that to all the registrants by email afterwards. And we'll also be having a Q&A at the end of the webinar. So hold your questions until then, and we can address them all at that point. All right. Well, thanks again for joining us, and I will leave it to Eileen.
Awesome. Thank you so much, Ron. So yes, today I am going to be talking about uncoding mouse affect through use of our novel judgment bias protocol. This is work that I co-led with Dr. Agustina Rosasco in the lab of Dr. Georgia Mason at the Campbell Center for the Study of Animal Welfare during my PhD. And as Ron mentioned, the detailed protocol is published and available on Jove. The QR code is here if you're looking for it right now. And it's also based on original work that we had published in Physiology and Behavior, where we had developed the task and then applied it to investigate the impact of subcutaneous tumors on the affective states of nude mice that are used in oncology research. So because the details are available in both of these publications, today I'll focus a little bit less on the nitty-gritty protocol elements, but more than happy to answer questions about it in the question period after. Instead, I'm going to talk about the importance of validation, updates and progress made in the task since the original publications, and highlight the ways that the task can be used. Oops, the mice I was talking about. So if you're here today, there's a good chance you already know what judgment bias is, but in case you're interested in assessing affect and you aren't familiar with the term, judgment bias is a type of cognitive bias, and cognitive biases are simply differences in the way that people in positive or negative emotional states process information. In humans, judgment bias is assessed through unconditioned responses to ambiguous stimuli, and it tends to refer to the way that people in negative states interpret ambiguous information as negative, while someone in a positive state might interpret the same thing as positive. So for instance, if I look at this panel of faces across the middle here, if I was feeling anxious, I might see that facial expression in the middle and think this man is not looking happy right now. But if I'm in a good mood, I'm feeling positive, I might see that exact same facial expression and think he looks neutral, maybe a little bit positive. And it's thought that these biases have adaptive value and are therefore conserved across species. And because of this, there's been some really thoughtful animal research operationalizing these optimistic and pessimistic responses to develop tasks for animals. Now, there's a range of approaches that people take when assessing judgment bias in animals, but the basic principles followed tend to be that animals will be trained that one cue predicts a reward, that another cue predicts a punishment or absence of reward, and then animals will be presented with an intermediate ambiguous cue. So for instance, if we train rats that a high frequency tone predicts reward, a low frequency tone predicts absence of reward, and then we present them with an intermediate cue, the expectation is that animals experiencing negative states will show more pessimistic responses than controls, as though they're expecting a punishment or absence of reward. And these tasks have been developed for a very wide range of species, from dogs to bees to cows. But surprisingly, for laboratory mice, which are the most widely used vertebrates in research, before our work, no task had been validated. And so I'm talking about validation a lot. So what do I mean by validation? Well, when I'm saying validation, I'm referring to construct validation here, basically asking, does the task measure what it claims to be measuring? And to validate a task as sensitive to changes in affective state, which is what we are aiming to do here, we first need to use research subjects who've had their affective states altered in well understood ways. Evidence for this can come from confirmed changes in other reliable indicators of affect or emotional state, or support from the literature showing that preferred or aversive treatments have been used. So we might choose to use housing or handling approaches that we know have positive and negative impacts on affect and welfare, or maybe a pharmacological manipulation so that we can alter state that way. For the task itself, we then have to demonstrate that animals can discriminate between positive and negative cues. Basically, can animals meet a learning criteria that we've set? And then we have to confirm that animals are interpreting the intermediate cue that we've presented as ambiguous. Here, we're basically checking, is the cue that we think is intermediate and ambiguous actually that way to the animals? And we haven't accidentally picked something that seems very similar to the positive or very similar to the negative for them, which is important because their sensory worlds can be much different from ours. And then finally, only if these three assumptions are met can we investigate whether affective state influences responses to ambiguity. So here, this is sort of the crucial point. A task would be valid if we can show that affective state impacts the responses in predicted directions. So negative affect would predict pessimistic responses, as though animals are expecting an absence of reward or a punishment, and animals that are in more positive affective states should show more optimistic responses, responding as though they're expecting a reward. But why is this so important? So validation is critical for any new indicator of affect, not just judgment bias, and well, any new indicator that we're using in research in general. And this is because if we don't have strong evidence that our indicators are measuring what we say they're measuring, and a solid understanding of the strengths and weaknesses of these indicators, then we risk drawing inaccurate conclusions. And this has pretty obvious consequences for research. If we're asking questions about animal welfare, it can mean that we're using resources to try to solve a welfare problem in a way that's really not helpful. We can mislead other researchers. And similarly, or sort of along the same vein, if we haven't laid out very clear requirements for validation and interpretation of indicators, we run the risk of harking, which is hypothesizing after the results are known. And this is where the aims or predictions of our work are slightly adjusted after seeing the results of experiments. And this can similarly lead to inaccurate conclusions, hinder evidence synthesis, and mislead other researchers. So quite problematic. If you're interested more in this topic and about validation in general, there's, tons of fantastic things written on it. But a recent BMC biology comment from Dr. Georgia Mason is a really lovely primer that, at least relevant to welfare research and assessing affect, directs you to some key papers and sort of covers some basic principles. So recommend looking into that. But the ultimate thing here is that validation is really, really critical, but it's especially important for judgment bias tasks. And this is because they're quite different from the human tasks that inspired them. So they'll involve operant responses for food rewards, which we tend not to do with humans. And we also often see with judgment bias that can lead to null or counterintuitive results. On top of this, for mice, careful validation is even more important because validating a task seems to be particularly challenging. So prior to our task, there had been 16 published attempts in the literature, and 11 of those had attempted to meet those 4 criteria and failed to meet 1 or more, so the task wasn't valid. And then a subset of them didn't attempt to validate it as sensitive to affect, so instead looking more at those key characteristics of can animals discriminate, and are we presenting animals with an intermediate cue? So valuable, but still not showing that it's a tool that others could use yet. So this is certainly something that appeared to be tricky to do, but we did set out to do it, and spoiler alert, our task was validated. So the approach we used was a scent-based go-go digging task. And here, go-go means that mice have to choose between making active choices during the task, so between one action and another. And these tasks have been shown to be more sensitive in animal judgment bias than no-go tasks, where animals have to decide between an action or refraining from a response. We also used odor cues and required mice to dig for rewards to take advantage of the natural abilities and motivations of mice. The trials themselves would look something like this. So during positive training trials, mice could choose to explore and dig in the scented arm. And this contained a pot marked with the positive discriminative stimulus odor and also a buried high-value food reward. You can see in the diagrams here, there's always an accessible compartment of the pot and then an inaccessible section at the bottom so that the treats would be equal across all. Or they could choose to dig in the unscented arm, which was marked with distilled water and contained a buried low-value food reward. In the negative trials, mice had to choose again between a scented and unscented arm, but here the scented arm now contained a pot marked with the negative discriminative stimulus odor and contained no accessible food reward, or the unscented arm, which as always was marked with distilled water and contained a low-value food reward. Mice would then be tested in positive and negative unreinforced trials, and those that had learned the task, we would assess them for learning criteria. Those who had learned the task would then undergo an unreinforced ambiguous trial. And here the scented arm would contain a pot mixed with a mixture of our positive and negative discriminative stimulus odors. So here a mixture of mint and vanilla, which presented our ambiguous odor, and no accessible food rewards. And then, as always, the unscented arm was marked only with distilled water, and these trials were unreinforced, so both sides had no treats available. So if we go back to our validation criteria, how did we show that this task was valid? So in terms of altering affective states, we had evidence that these were altered because we differentially housed mice in either large, well-resourced cages, like the ones up top here, or conventional shoebox-sized cages like the one shown below here. And there's a large body of literature showing that these have impacts on welfare and affective state, the large, well-resourced environments having a positive impact on welfare and affect. We then showed that mice could discriminate between positive and negative cues. So we had set a learning criteria that mice had to dig for twice as long in the positive trial as they did in the negative test trial. Which they did. And then also I have plotted here just their latencies to dig and their digging duration, so you can see the differences between the positive and negative trials. We also confirmed that the intermediate cues we presented were interpreted as ambiguous. We had this nice graded response that we were looking for here. And then finally, the crucial piece was that we demonstrated sensitivity to affective state because animals showed the responses in the predicted direction. So our conventionally housed mice, the ones that were in those shoebox-sized cages, had longer latencies to dig in the ambiguous trial as though they expected the absence of reward. So since that original validation, we've also run a replicate experiment, and we sought to make a couple of small changes to help streamline the task because our task, well, judgment bias tasks are notoriously labor intensive. Ours actually didn't have that many trials per animal relative to others, but still is a lot of work in the lab, which limits the number of animals that you can test at a single time. So we're hoping to make some subtle adjustments to help increase this and make it a little bit easier for ourselves and then for others to adapt. So if we're looking at the full experimental timeline, again, we differentially housed mice in these conventional or well-resourced cages. And here we used the same mice that we used in the original experiment, so C57 black 6 and BALB-C females, and then an additional strain of DBA2 mice. Our digging training phase was the same, and essentially this was a section of training where mice would be offered positive trials and treats progressively buried over the course of 5 days so that they would learn that they had to dig in the pots for buried treats. And then during our discrimination training phase, this yellow block here, This is where mice would be presented with positive and negative reinforced trials so that they would learn to discriminate between odor cues. Originally, these trials were 5 minutes each per mouse, and each mouse would have 4 trials per day. Anecdotally, in the first experiment when we were running it, after the first 1 or 2 minutes, mice had sort of performed their response and then somewhat lost interest in digging and were exploring other areas. So we decided to reduce this time frame, instead making these trials 3 minutes each. It sounds like a subtle change, but 2 minutes per mouse really added up. And so this actually, I think, ended up being really impactful and allowing us to test a lot more animals. And then in the testing phase, in the original work, we had tested mice. So during the week of testing, on the first day, they would have their training trials and then either a positive or negative unreinforced test trial. On the second day, they would have the other one that they hadn't received. So if they had day 1 positive test, day 2 would be negative test. If they met learning criteria, we would then test them on the ambiguous trial. In the original work, if they hadn't yet met learning criteria, we would continue to test them for up to 5 days to see if they had met learning criteria and then test them in the ambiguous trial. In this replicate work, we simplified it and just did the 3 days. So mice receiving positive test trials day 1, negative test trials day 2, and then those that had met learning criteria received the ambiguous test trial on day 3. The reason that we did this was because in the original experiment, only 2 or 3 mice that hadn't met learning criteria in that first sort of wave ended up meeting it with the subsequent training. So this just really simplified things, and we felt like we weren't going to lose that much with the approach. So in terms of outcomes with these subtle tweaks, it really, really improved our efficiency. So when we were training and testing mice, we did the experiment in 2 batches, but we were able to train and test up to 60 mice at once, having only 2 researchers, which was a pretty big jump because our original experiment, we had trained and tested 35 mice at the same time with 3 researchers. So this really allowed us to have a larger N in the experiment. And very importantly, this didn't come at a cost to successful learning of the animals. So these slightly shorter trials and that simplified testing phase, we had 85% of our mice meet learning criteria. And in the original experiment, it was 88%. So we were still quite happy with that. The mice still seemed to be learning the task effectively. And then, of course, importantly, we still found it was still sensitive to effective state. So our well-resourced mice or enriched animals showing more optimistic responses to ambiguous cues. This was true for the original 2 strains that we had used, those C57 Black 6 and Balb C females. But with our additional strain of the DBA mice, these are preliminary analyses, I should say, but we currently have a non-significant housing effect for them. So future work should certainly investigate strain differences further. But taken all together, what does this mean? So to our knowledge, this is still the first or the only validated judgment bias task for mice. I've kept a few more tasks have come out. I have not seen one that with construct validation yet. However, if anyone is aware of one, please feel free to send it in the chat. I would love to read it. But these are incredibly valuable tools for assessing mouse affect. So testing animal welfare hypotheses. We wouldn't recommend using these tasks for day-to-day welfare assessment, but if we're trying to make decisions about different types of husbandry or housing, this can be an excellent way to get at some of these subtle changes. And it also opens the door to many other diverse research areas for investigating the links between affect and the diseases or conditions that we're using mice to model. So for instance, the work that we did in the original physiology and behavior paper, assessing the impact of subcutaneous tumors on mice, things like that. Are there changes in affect that could be detected before other clinical signs? Again, it was easy for mice to learn, which we think is likely potentially due to the ethological design and humane approach. But the fact that it's easy for mice to learn hopefully means it's also easy for others to adopt because of that high success rate and ability to test many animals. And so in terms of what the next step should be, we certainly still need further replication and refinement. So judgment bias tests are useful for assessing a diverse range of stressors. Currently, we've been using this to assess chronic stressors. So future work should look into whether it's sensitive to acute manipulations, also looking into the use of near positive and near negative cues, looking into retest reliability. And anyone doing this, we recommend running pilots to confirm treat preferences within your mice to help keep them motivated and to identify ambiguous odor mixtures. So to make sure that your cues that you're using, particularly that ambiguous cue, is interpreted as intermediate as you expected. So with that, I will just say thank you so much to Joe for organizing this event, for the funders of this work, to the Mason Lab. A lot of hours went into these experiments, our animal care staff and our mice. And I will open the floor to questions.
All right, thank you, Eileen, for the excellent presentation. And like you said, you know, if folks have any questions, just add those to the Q&A. And we will be sending an email afterwards, so you know, if you think of any questions after the fact, feel free to respond then, and we can address those as well.
Okay, so we have a question here from Brianna Gaskell. Hi, Brianna. We mentioned evaluating treat value. Did we assess reward preference for each mouse? So we had done pilot experiments prior to the original work where we offered mice a range of really diverse treats, which was lovely experiments to be running. We offered them everything from like dried mealworms to sprinkles to freeze-dried chicken. And it ended up being that they just really loved dried sweetened banana chips and almonds, which most people are, well, almonds are incredibly common rewards, so that was good. So there were some individual differences though. So one thing that we did actually in the replicate experiment, which I didn't mention, was we added, our original work had used the dried sweetened banana chips. But then we also had so we would have a banana chips and an almond sliver in the pots in case some of those individual differences were altering motivation during the task. We figured having both available would mean that if a mouse really preferred the bananas, then they had it there. If they really preferred the almonds, then it would be there too. And they're small, so it didn't take up a lot of space.
These mice have a better diet than I do. It's pretty nice to have.
Well, their normal diet is like the hard rodent chow, which they get day in, day out. And so that's the other thing. I think they're probably quite
It's a
motivated.
change. Yeah,
Yeah,
exactly.
yeah.
Okay, so it looks like there's no other questions. Oh, one more.
Oh, speculations for the DBA strain being not so good. This would just be anecdotal on my part at this point, because those analyses are preliminary and haven't had a chance to dig into them yet. But the DBAs, they tend to be quite active. They're within the cages, both in our enriched and conventional cages. They're moving around a lot. They also are sometimes perseverative on tasks, so they're prone to stereotypic behavior and perseveration. The thing that we noticed was that they just seem to be incredibly motivated to dig during the task, almost regardless of not regardless of what was available, because they did meet learning criteria, but they were just very fast each time to go either to the positive or to the negative trial. So possibly a sensitivity issue because they just were running there right away, digging really fast, getting really into it. And so, yeah, I think their strong motivation to dig may have actually been a little bit of a hindrance there. I see another question from Brianna Gaskill. So when using scent, I always worry about scent transfer onto your hands. When baiting the pots or the cues, did you do anything to mitigate this? So that is a great question. And yeah, working with scent is tricky, and we got so many questions about this in terms of times when we had run the task. I'll pull up this slide here. So when in the arenas, we had like a slotted dispenser that would have a cotton pad that was marked with the odors at the start of each arm, and at the end of each arm, so within each pot, so that, and the bedding itself would be marked with the scent. So I think absolutely, like, mice have such strong olfactory abilities that I'm sure as soon as they were in the room at all, they were probably getting both scents. The difference would have been that those cues that were right in front of them in the arms would have been so much stronger. So I'm sure they were picking up on subtle differences and smelling different things throughout the room, and throughout the arena, but those cassettes at that very front would have been much stronger, and it seemed to be enough to keep them learning the task effectively. So presumably not problematic, because if that was the case, I think we would have had more trouble with mice meeting learning criteria. But yeah, it is a really tricky thing to control. And that was another thing I'll just mention that we had thought about a lot of in terms of the treats, because mice do have such impressive olfactory abilities. We were worried about having them actually just just know which treat would be available in each pot based on odor alone. So we had each pot would contain it had the accessible area where filled with bedding, and if there's a treat available, it would be buried there. But then there was an inaccessible compartment that was perforated, so that would have the other treat. So every single trial would have the same treats available. So presumably the arms always smelled the same from a treat perspective. But yes, the sense and the olfactory abilities of mice are something that was on our mind a lot.
All right, well, thank you everyone for the excellent questions. Looks like that's about it for the Q&A, unless anyone has any last minute ones. So otherwise we will send around an email of the recording in the next day or two. Feel free to respond to that if you have any questions, or if you are interested in publishing anything similar. We can discuss that as well. So yeah, thank you again, Eileen, for excellent presentation, and thank you to all the attendees.
All right, thanks so much.
All right, have a great day. Bye-bye.